experiments funded by DBT on breast cancer. And currently we are also running jointly a national network project on breast cancer. So he has trained many of the students from Mizoram University as well. So he is a very good scientist, and I'm sure all of you will enjoy his talk. Sir, please. Yeah.
Thank you very much, sir, for kind introduction. And I'm really honored to present first talk here. And yesterday I really enjoyed so much diverse talk, and it was very informative and knowledgeable. So since I'm coming from cancer hospital, so I request organizer give me 1-2 minutes extra so that I talk a little bit about the cancer to have no cancer. My talk title is Proteomics Studies set biomolecules, a set of predictive and prognostic ECTREC. My institute name is ECTREC, Advanced Center for Treatment Research Education in Cancer. And if you see, there are two wings. One wing, this is called CRI, where scientists work, and this is a wing that called CRC, where clinicians work. And between both, there is a bridge. This bridge physically exists, and the purpose of this bridge is to have a better communication between the scientists and clinicians. So, I like this statement very much. We have too much high throughput, and with the help of high throughput, a single laboratory can produce more bits of a data. Listen, more bits of a data than Shakespeare managed in a lifetime. So I'm talking about data. What they are saying, data, not results. So now this is time we should focus on the results also. See, if you see the cancer, after cardiovascular, this is the second most deadly disease. But cancer hospital, ground for the cancer, everybody is talking about the cancer. Why? Because this is one of the deadly disease that put tremendous burden on patient, family, and society. And this is the statistics. By the year 2030, this will rise in expected 26.4 million cancer cases, 17 million dead, and 75 million people living with this disease. So does anyone want these statistics? Nobody wants. That's why I cut this line, because we don't want this statistics. But do you think cutting line is a solution? No, cutting line is not solution. This statistics will be there if our ability to prevent, diagnose, or treat cancer does not improve. Yesterday, Honorable Vice Chancellor was saying physical parameter are well defined, mathematical parameter are well defined. So things are easy. But biological system, understanding biological system, it's really very difficult. We have gone to the Mars, hitting other planets. But one biomolecules virus, this SARS-CoV-2, he has not even allowed us to come out from the home target going other planets. So understanding biological system is very difficult. You see the incidence of a lung, it is number 1, and still mortality is also number 1. But if you see the breast cancer, number 2, and if you see the mortality, number 5. That means some success are there. There are far more to go, but success are there. And breast cancer is treatable if prevented, if detected early. It's treatable. I'll repeat again. So what is my laboratory is doing? We start from the genomics, and rightly Dr. Sethil pointed out, we have one BRNS grant running on the breast cancer mapping sequencing on the meso populations, and I'm not talking about the genomic parameter because I will be talking about the proteomics. And then we start from the genomics, go for the proteomics, and then structure biology and bioinformatics, a long journey. We all do together and cancer is not one stem disease, it's a multi-stem disease treated with the chemotherapy, surgery, and radiation. So similarly, one person cannot handle unless until you don't have interdisciplinary approach. So genomics parameter, I will just only touch 2 slides. This is the BRCA1, BRCA1 genes has been discovered in 19 is discovered in 94. 34, close to 30 years back. And what has been identified? Several variants, and these variants have been identified throughout of the BRCA1 genes. And new variants is coming out. All right, it is very easy to say that variants are coming out, new variants are coming out. Now the challenge lies to the scientists and clinicians how to categorize pathogenicity of variants. Which variants is responsible for tumor formation? Which variants is responsible for the cancer growth? If you see the challenge, I'm planning to crystallize and determine the structure of these genes with the binding partner, not only me, across the world. So far, structure detail only this BRCT is reported, and only NMR structure of the ring domain is reported. Rest of the details is not available. So it's not only people across the world are working, but still there is a problem in purifying the protein, crystallizing, because of this disorder region of this protein. So just wanted to tell the young people that to work on the cancer, you don't have to work the cancer hospital. There are all databases available. If we identify any genetic variants, we have to provide the details to the database. Sitting in the Mizoram University or sitting anywhere in the corner of the country, you can explore and get the details. There are several genomic databases available, and for BRCA1 and BRCA2, close to 1600 genetic variants has been identified. 1600. And if you see, out of 1600, what is the statistics? Only 2% are the deleterious, and 5% are identified as a okay the pathogenicity. So the challenges are big challenges are there, and we are getting several now sequence variants are getting from the different populations, and that we need to still categorize. See, this is the one of the. With this, I will stop talking about the genomics. This is family pedigree. Please, this message for young people. This is the family pedigree. First generation, second generation, and see third generation. One single mutation, C16972Y, one single mutation. It's very easy for the presentation. For the slide wise, it's very easy. See, 1, 2, 3, 4, 5, 5 sisters. How many are affected? 1, 2, 3, 4. Okay. So, from the sequencing from the Tata Cancer Hospital, these are the variants what we identified, and our finding has been correlated with the IARC, International Association of Research in Cancer. See, these are our finding, and our finding is saying they are classified as class 5, and that is from the matching with the IARC. And if something is classified as class 5, they are the high risk. So these are the surveillance. So this is how. So this now the question is, what is the translation? Now it has been translated into the practice of clinical oncology. Genetic counseling is started in different part of the countries. Of course, more has to explore. So these are the parameters that we have categorized. So this is just wanted to be brief about the genomics. Now I will talk about the proteomics. That is my title. I am working on the proteomics of HNSCC. Set up proteomics. You see, we cannot work on one molecule because HNSC, head and neck squamous cell carcinoma, is oral cavity, tongue, larynx, pharynx, nasal cavity. This organ, if the cancer is in this organ, we call HNSCC, head and neck squamous cell carcinoma. Okay, so one molecule is very difficult to handle, very difficult. And this is current. What is the title? It is fully matching with the seminar current trends in the biological sciences. So the role of mass spectrometry is starting. See, who is responsible for this? 75%. You remove this, you go in the Tata hospital or different hospitals, you will be really surprised to see the crowd. There is no, we don't have proper management. But if you remove this 75%, very good. There is a, of course, only this is not a one factor, but see, this is the how what factor is very seriously responsible for this. And besides progress made in the couple of decades on head and neck cancer, treatment protocol has been standardized. So much has been done, but the survival rate, 5-year survival rate remains below 60%. So it's challenging task. We have to identify, either you identify early detection biomarker. I'm not a clinician, I cannot standardize the surgery protocol, but prevention and early detection prevention, if we can work on that, and that will be very much beneficial. So that's our work. So our aim is to identify a panel of a differentially expressed protein using a quantitative serum-based proteomics platforms. So we will be working on the serum-based. All right. So what is the problem with treatment? Late presentation of a disease. The disease is inside, is not appearing. It's challenge. It's not that some disease is. So this is the issue in the treatment. Late presentations, presence of occult metastasis. Occult means hidden metastasis, and high rate of relapse and recurrence. So we need to improve the method to detect occult metastasis, early diagnosis for the recurrent, and so that challenge is to identify and validate the robust, predictive, and prognostic biomarkers. So what are the biomarkers? Just one slide. Diagnostic means identify the type of a cancer. Predictive means that predict response to the given treatment, and prognostic means provide information about the patient, what is the overall cancer outcome. So there are different kind of a nucleic acid, protein, all biomarkers are important. All we cannot work on. Someone is working on the nucleic acid biomarker, it's work to study. Someone who is working on the proteomics biomarker, it's work to study. So, but protein, see what happens, gene expression level do not always correlate with the difference in expression of, at the protein level. So that's why we decided to work on the protein level. That may provide better understanding to disease process as well as help in the treatments. So now we can ask the questions. This is the serum. Serum is coming out from the blood, and blood is the body, a kind of circulatory body fluids. So we have to correlate with the tissue also. There are some biomarkers, but their sensitivity in specificity is not as better as we can nicely correlate. Okay, these are the differentially expressed proteins, so we can standardize the treatment protocol or predispose someone. It is not that level. So that's why there is urgent need to identify and validate protein biomarkers which can aid in the early identification of the disease and help in the clinical management of HNSCC. So now what is our now we recruited 50 patients, okay, and now I want to recruit 120 more. So someone was saying, I wish you don't get any patients, don't. So that's fine, yes, but I want to recruit. So this is the result from the 50 patients, and there were the 50 male, and this is the tumor site, and this is the tumor stage. And what happens to the HNSCC once patients report to the clinic? There are a disease management group, DMG. In the DMG, surgeon is there, radiation oncologist is there, medical oncologist is there, scientist is there, and disease management is group over there. So after surgery, he will go to the radiation oncologist or he will go to the chemo. So radiation oncologist will tell him to go for the pathology. Before radiation, he will go for the pathology. Pathologist will take 5 ml blood. With that blood only we will take a 500 microliter blood. We are not doing extra injection to the patient. Our study, our ethics will ask them several questions if you do all extra stuff. We will take only 500 microliter blood from there. So before, please listen, this is before radiotherapy, he will go for the pathology. After 48 hours of radiotherapy, he will go for the pathology. And after 1 week of radiotherapy, he will go for the pathology. And this is the post after 1 week of, 1 month or 2 month post. So we have taken the blood sample, 4 time point. Pre-radiation, 48 hours of radiation, 1 week of radiation, and post-radiation. And from this blood, we have taken the serum, extracted the serum, and done some mass spectrometry. What kind of mass spectrometry I will tell you. So this is the study. With this, we also have a correlation with the survival rate. This 50%, how many are surviving for 5 years with the given treatment? So those who are surviving, they are good responder, and those who have not survived, died, or relapsed, something happens, they are the poor responder. So this is interdisciplinary. We have a finding from clinics. Okay, we have taken the blood serum of this patient, and let's see what is the response of a patient of this given treatment. How many year he is surviving? 2 year, 4 year, 5 year. So we have a correlation. The presentation what I am showing, it's not a 1 month. It's a correlation of a 5 year survival rate. So from this, so what we found, 31 patients were good responder and 19 patients were poorly responder. Means something happens with them. So now, I have designed the experiments. I want to do the mass spectrometry, and sometimes, people say technology should not be problem. Technology is the problem. Unless until we don't have technology, how we can identify how can we receive our goal. So my student, what I wanted to tell you to all the young people is that having good student is really blessing. Of course, having good guide is also a blessing, but having good student is also a blessing. So what was my purpose? I don't wanted to do any gel-based study. I just wanted to take the serum and shoot directly to the mass spectrometry to check what is present. Let's see what is present, what's coming out. Then I discussed with my students, and then in the serum there are highly abundant proteins, high molecular weight proteins, and they are present in high amount. So the purpose is, first purpose, to remove those highly abundant protein and enrich low molecular weight proteins. So many commercially available screens, everybody is saying, after spending huge amount of money, she started doing all the screens, and then we were failed. Means highly abundant protein were going out, but not in that way, not that amount that mass spectrometry can recognize low molecular weight proteins. They were masking in our predictions. So, so this become one challenge. Going to the further analysis was fine. We could not shoot serum directly. See, all number of kids, and you see, we this is a before depletion, means before passing blank, and then flow through after a kind of a from the serum. I think everybody knows, and then beads after binding, and protein, that the protein markers. And when we loaded this, we wanted to analyze this low molecular weight proteins. When we loaded onto diesel, we found only few proteins. There are not enough proteins. Then it's after spending huge amount of money. Then what happens? See, through gradient gel. So in the gradient gel, different molecular weight proteins will disappear, and then low molecular weight proteins comes out. She started doing this, and this was the successful. But again, it is in the gel based. Then what is the new gel based? Everybody in gel based, something is poorly expressing or poorly present, very difficult to detect it out. Then the purpose will not serve. Then of course this gel based work well. I'm not saying, but the purpose, what was the purpose? To directly shoot, it was not serving. So gel filter method, except it was excellent. Then she has further done aggressive studies, and she found that acetonitriles, a different concentration of acetonitrile at different temperatures, deplete highly molecular weight protein and increase low molecular weight protein. So see, after spending huge amount of money, we were not getting, in this protocol we got, and see all the most of the highly abundant protein depleted, and this is the result of a 2 So this itself took her 2-3 years. But students are time bound; they have to submit. But it was good. She has standardized, and we were in a position to deplete highly abundant protein and enrich low molecular weight proteins. Then this is the protocol. So what we have is, this was a very expensive mass spectrometry technique is very expensive. So 50 patients we cannot do individual 1, 2, 3, 4. It will huge amount of. I approach to the company fellow, they say 10 sample 10 lakh rupees. So 50 sample 50 lakh rupees, everything is gone. So see what we have done, we have pooled the sample. We have pooled the sample, and from the pooled sample we started analyzing. So good responder pool pre-RT, good responder pool 48 hours, 1 week post-RT, and then poor responder. So this is the protocol. So this is, I will share the slides if needed. This has been published. So see, I track labeling from the, when all these kits were failed, we were identified 90 with the 95% confidence protein close to 180 to 250. Then this has been published. The student has to publish. We don't want it to go for patent because let's publish fast. Then whatever we identified, let's validate them. I will take some time here to help you out to understand the protocol. So we were having 4 time point. In 4 time point, all were good responder, and someone were good responder, someone were poor responder. So what we have done from the pool, 50% of the pool, protein level in poor responder were determined using good responder as a control. Pre-RT, before RT. Poor responder has been taken considering good responder as a control. Then in another objective, pre-RT sample. Pre-RT sample were taken as a control and rest were the reactions. So this was the discovery level. Then from the 50 pool, we randomly selected 10 patients. 5 who were good responder and 5 who were poor responder. So what is the result from the pool and what is the result from individual? I'll help you out to understand. So this is the protocol. If you see this protocol, so it is, see, we are sometimes feel ourselves privileged to present the work of a student. See, we spend huge amount of the time. Sir is knowing better that to standardize these protocols. So, but having good student is a blessing because of her. Sorry. So, how many 180 to 200? All these proteins, everything. She has to identify each protein and correlate from the database. What is the relation with the cancer? What kind of tissue or space is associated with these proteins? And then after doing aggressive, see, I will be simply by click change the slide, but to come from this slide to this slide, she took longer. And she identified 14 proteins. Those were, this is the sign for down regulations, and this is the sign for up regulation, and this is the base. So out of 14, see, clustering is differentially expressed. And this is from here, this is the first reaction when we have taken poor responder considering the good responder as a control. First up. Okay. Then after this, further we have taken pre-RT and then post-RT in a different time frame. This is the protocol for that. And we have concluded, we found 10 proteins from the reaction. Pre-RT, 48 hours, and post-RT, and 1 week. Actually, we have, if we are identifying 1 week or post-RT, then it is not early detection. The purpose is to identify soon after the treatments, 1 week or 48 hours. So that's why we have not taken those stuff. And then these are the good responder and these are the poor responder. 48 hours are how they are down regulating and 48 hours are how they are up regulating. See the clustering with the good responder, it is down regulating. With the poor responder, it is up regulating. So clustering, these other proteins, gilsolin, these other proteins, they are differentially expressed. Okay. So
from this, we decided to, this was the from the pool. Then we decided to go further. This is a kind of a, people is saying Western blotting can also be done. Western blotting, you need to identify first protein. So mass spectrometry, when a very small amount of a protein, even it is present, can detect nicely. So the purpose was to use only mass spectrometry. So that's what we have done. So in the validation phase, we have to perform targeted proteomics. So now we need to identify what kind of a proteins are expressing there. So that is called parallel reaction monitoring. This is just one is basic information that we have identified through the parallel reaction monitoring. And if you see, these, we have taken 20 proteins, and from the 20 proteins further correlated and analyzed after a place of literature search. See, this is huge amount of data. It's very easy to change the slide. In each slide, a lot of the work student has to do. And then what has been identified? See, the first pre-treatment expression, when good responder was controlled, clustering was differentially expressed. And when there was a reaction, 48 hours, 1 week post-RT, see, the clustering was differentially expressing. It is in down-regulating with the good responder and up-regulating. So we have to do, this is the study in individual patient. We randomly selected 5 good responder and 5 poor responder, and our study is saying whatever has been done in the pool, the same is available in the individual patient also. So you see, clustering, pre-treatment upregulation of clustering in poor responders indicates that patient may develop, they are the poor responder. And this is the upregulating, that indicated may develop aggressive tumor in a coming time. That's why they are the poorly responder. So we have done the study, literature search, and from the literature search, we found that, okay, yes, we are going in the right direction. Similarly, gelsolin. See, this is the finding of the gelsolin. And gelsolin, this is from when good responder in the baseline, and with the good responder, when, poor responder when the good responder was the control, how that is gelsolin is differentially expressing. And this is with the individual. And when we have done the, so pre-treatment upregulation of gelsolin in the poor responder seems this patient can develop, that's why they are the poor responder. This is the literature search from the good responder. We have gone through and then found that we are going in the right direction. So this is the individual patient. From the pool, we have selected individual good responder and individual poor responder. So now, so this is the summary: 1.5 to 2.4 pre-upregulation of clustering gelsolin and other proteins, and 2 to 5-fold upregulation of within 48 hours. So this differentially expression of a protein can be a very good biomarkers which can correlate. And see, I told you that it's not a 1-2 years. We have a correlation of a 5-year survival rate. So this is the result of a 5-year survival rate. But this panel of a 12 protein biomarkers can help. Now the challenge and question to the students and everybody, I'm very much open. This is the serum proteomics. And serum is the circulatory body fluids. So reviewer is asking, why can't you focus on the tissue? Right? It is very easy to focus on tissue. It's not as easy as ends. Okay. So I wanted to populate this. The reviewer is saying you have taken 50 patients from the pool, and this is a 10 from the 50 only. So definitely you will get this. So you guys are understanding now. This is the 50. I have taken 50 and then selected. Definitely you will get this. Now you do a study from independent cohort. So 1.5 years back, since that I submitted my ethics last month approved. So I'm taking 120 patients further and then further validating. And then once it is from the blood, we will go for the tissue specific. And hopefully if tissue specific is valid, then definitely there will be very good biomarkers. So this will allow to design better treatment strategies, help to tackle problem of metastatic and other. And this is my group. funding from DBT, DST, ICMR, DAE, and the girl who has done more proteomics work is this, Lipidar, and all these are different members. And does anyone know this girl? She is Dr. Rishta Barua. She was a postdoc in my laboratories. And find out who is Dr. Rishta Barua. I'm letting you guys to know. And these are 3 students joined. So besides, as an SCC, I'm also planning to work on the urine proteomics. So we have proteomics. Thank you so much. I will be happy to. Any question, please feel free. And cancer is treatable. Don't need to worry much. Detected early. But if this fellow is, he hide inside the body. So that's the challenge out. Someone is hiding at home. So that's why different kind of a weapon is needed. And yes, someone is coming with arm, we should have arm. Someone is coming with missile, we should have missile. So of course, it's a really very deadly disease. We all have to be very careful for this. Please feel free to ask. I stop my time fast early so that I can have interaction with. Please, sir. Thank you so much. That nearly 12 proteins we were talking for the evaluation. Can we develop a kit for easily detecting or screening type of thing? Right. Is under the process. That's the process. Once I validate to the independent cohort and then go for the tissue, then we can Western for card test for response. Yes. Then it will be mass spectrometric and not done it. Once it is validated, then yes, it can be done. It can be done. Yes. By developing antibodies. Yes. Smaller. Yes. So this is under study as a proteomics. We have done serum proteomics. Now independent cohort need to do, and then tissue is specific. And with this pathway, I'll later we will do. Good. Yes. Thank you. Please, sir. Okay. Pardon me, sir? Developed by Extract last 8 years. Indeed, drug developed by Extract last 10 years. Sir, yes, there are so many, some things that has gone. What I remember, one viral biomarker was developed and it was gone to up to the drug controller of India, and then something happens. I don't know much. But what I can tell you, our genomic based approach, this is not, I'm saying me, team. And we have clinicians and we have collaborators that has been integrated to practice of clinical oncology. Genetic counseling is started, surveillance is started, and people are getting benefited now. And of course, we need to explore much more so that people can understand. Most of the people just think it's cancer. Sometimes, yes, right now this is not covered by the, and this is a little bit expensive, and of course the price is going down. And you see our patient clinicians, right? And they say, "Sir, मेरे पास तो खाने का पैसा नहीं है, आप medicine है. Sir, I don't have money even to go home." And then, so these are the challenges in our country. We all have to tackle it, but those are the challenges. Other sides, as a scientist, we all have to work on this. Yes, right. Man, please.
It's not a process.
Please.
Simple thing also the
Right.
but when we went for the testing, it did third level. So this is a common suggestion from yours.
Sure.
I'm asking for everybody. So every time we are observing, whenever patients are going for any test, cancer is on third level or the last stage. So is there any test which everybody can go so that cancer can get it in any stage?
Sure, ma'am. It's a very good question, ma'am. Actually, there are different kind of the cancers. Breast cancer can be detected early. and can be cured. The woman will not die because of a cancer. She will die with somewhere else. It's curable. Some cancer are highly grade 4. For example, brain tumor. And I the challenge for the students, you all are brain tumor. This is a brain and tumor is covering nicely to the brain. Now surgeon performs surgery. Can he touch the brain? If he touch the brain, the patient will die because of this, not because of a cancer. So he will remove the tumor, leaving some tumor inside, and then put the and now this tumor can further grow. But what can be done? The pain what he get that can be reduced, and life is spent little bit 2 months, 3 months. So surgery 2 months, chemo, radiation 2. So grade 4 can survive close to 1 year, 6, 7, 8 months. Some survival rate are better. So depending upon what kind of a cancer is there, it's all the cancer have different etiology. But no need to bother. Please be happy, healthy, eat nicely, and sleep nicely. Nothing will happen. Sir. So our proteomics, these very good. As a scientist, these proteins are differentially expressing. Someone are upregulating. So now what is our standard? It may, whether clinician listen or not, we can advise them that this much radiation dose, because of this much radiation dose, this protein is differentially upregulating. And this it is upregulating, survival rate are this. Can we reduce so that we can increase survival rate more? Long way to go. But answer to your question, these proteomics differentially expression proteins are predicting that because of that they are poorly responding and some are good responding. Students, that's what I was. Thank you so much. I thank Dr. Senthil, Then all the faculty member, and thank you very much.
Thank you very much, Dr. Verma. So as a token of appreciation, I request Professor Senthil to hand over a certificate to Dr. Verma. Thank you, Dr. Verma, for all your contributions to our society. I request Professor Senthil to remain on stage to deliver the vote of thanks.
Yeah, thank you, sir, for your excellent and innovative talk. So I would also like to add on from the introduction that we gave. So we have also followed the same protocol what sir had, Dr. Ashok had done his proteomic study. And here also we work on head and neck cancer, and there also we have found very good markers. Even in the breast cancer patients as well. From the Mizo population, we have identified very good markers for early diagnosis and a good prognosis, of course, with Dr. Verma's collaboration. And we are also in the process of going into the genetic counseling and bringing the markers into the clinic. So we are also, with the help of Dr. Verma, we are also trying to translate our findings. So here, not only thanking him, but also on his behalf, I would also like to say that he is open to collaborations. Anyone who is interested in research, he will train in his lab for 1 year also, any student or any faculty, which he has been doing for I know him from 2011. And so far, from the Northeast alone, he has trained more than 500, 600 people at different levels. So anyone who is interested in working on cancers, especially on genomics, can approach. Thank you, sir. Thank you.
So now it is time to transition into our 2 concurrent sessions again. Similarly, For Group A, please remain here in Hall 1. Group B, Plant Sciences and Biotechnology, we will be moving down to Hall 2. Sessions will begin shortly. Thank you.
I'll introduce you and I've got a very long list describing Dr. Jayaditya. Did I pronounce your name correctly? All right. Okay, I'll try my best to summarize this. So, Dr. Jayaditya did his master's in bioinformatics and later on established the NGO known as Help Earth, all right, which has been active in doing conservation work in Northeast India. And he's also active with many other organizations, collaborating with other NGOs, and has been receiving a lot of international grants, which is kudos for him. And he's also a member of the IUCN SSC Tortoise and Freshwater Specialist Group, and also the IUCN SSC King Specialist Group, the Amphibian Network of South Asia, and Indian Birds Conservation Network. So he has a number of publications, and many of which are being referred to by internationals. And also, he's serving as a consultant on various aspects of biodiversity conservation in Assam. So, without further ado, may we request Dr. Jayatidya to talk about his Blue Mountain Chronicles, Herb Havens and Hidden Wonders. Thank you.
Hello, can you hear me on? I hope so. Good morning. So, there has been a lot of technical sessions and lot of in-depth scientific things has been told. So I will keep it very light, and it is just a story that I have to tell you regarding this beautiful land of Mizoram and the amphibians and reptiles that we have encountered in this place. So this is a last minute presentation that I made. So generally, if you are defining a presentation, I sometimes say that presentation is something that the presenter writes by spending sleepless night to make the audience sleep when presenting. So I hope I do not do that. So this is like more like a story of whatever has happened throughout this my collaboration in Mizoram and whatever we got and how it all started. Next slide. I can do the next slide, or should I? Okay. Okay. So I just jotted down some of the things regarding Mizoram. Most of you know that, but the most important thing that I want to highlight here may be why amphibian, because most of the northeastern state I have traveled and looked at amphibians and reptiles in the classes of taxonomy. Like, I'm not good in ecology or anything else, but mostly taxonomy, like which is related to how they are related to one another and what are their phylogenetic relation. So one thing very interesting comes out in Mizoram is that why Mizoram is having so much of diversity. And I'm very happy to tell you, like I'm really fascinated by the fact that all the amphibians and reptiles, even birds and all, they have local names. In Assam, we the common amphibians and reptiles, we do have these local names, but for rare amphibians or the amphibians or reptile deep inside the forest, we do not have that local name. But in Mizoram, I see every amphibians and reptile, most actually, they have these local names, and that is fascinating for me because that has a conservation thing associated with it. When the community, it is not about scientist or researcher knowing about the species, and we cannot do conservation that way. But if the local community knows about the species, the existence of the species, that is more important. And local name for me is one of that tool which helps us see the community's outreach towards that animal or plant. And also the biogeography, how the Mizoram is built, the elevation, generally the elevation is like 1000. I think in Mizoram, Aizawl, it is like 700 meters, somewhat like that. and the Blue Mountain Peak, from which we say that land of Blue Mountain. Blue Mountain Peak may be something around 1700 to 2000 meter. So, and then you go to Kolaship and then Vairinti, the borders of Silchar, it goes down. So there is a huge gradient coming up. And maybe, and also apart from gradient, because Meghalaya is also having gradient. But the temperature in Mizoram is different from Meghalaya. Though that there is a variation in altitude, temperature is not as cool as that in Meghalaya. So that is also because herbs are, cold-blooded animal, and they do not do good in those cold climates. So that I think all those clubbed together makes it a heaven for, amphibians and reptiles. So just for the students, I know there will be people from different fraternities will not having, I may know, but for a generalistic, different fraternity, I need to tell you what are amphibians and reptiles. So amphibian constitute of toads, salamanders, Sicilians. These are amphibians, and then reptiles, snakes, lizards, amphibians, turtle, crocodile, and tuatara. Now we can leave this tuatara and amphibians we do not get in India. But rest we do get in India, and for also Northeast India we get all those things. And I'm very happy to say that there is a revival of gharial population in Assam. Assam at one point of time we were devoid. We did not have much records of gharial, but now slowly we are having a lot of sightings of gharial. But here we have this amphibians, all the groups we have in Mizoram as well. Yesterday, Decemson, when he was talking, he was showing this salamander as a new species that we have described from Manipur. So, just because I was telling you about the stories of Mizoram and how it all started. It all started in around 2014, and that time it was not so easy to come to Mizoram, and road were not that good, and we to save money, I had to come through jam-packed sumo from Guwahati to Mizoram. It's a lot of journey to travel, and then somebody is vomiting here and somebody is vomiting there. That we also have to take in account. And but the pain that we took is worth it, and we have made friends for life here. That's more important than any other discoveries that we have done here. So you see, this is a cartoon representation of us. This is the person, one of my very, very good friend, VL Shrima. He's not here, I think, this time. And he is a great person. And this, I am like taking a photograph of this coral snake. So, like to give you a context of how rich we are, where we stand in the history of amphibians and reptile. If you see, this is a global data of amphibians and reptiles. This side you have amphibians like 8223 species, and these species go up and go down because many new species are discovered, and many new species which were discovered get synonymized because of some reason or so. And you see reptile 11,341. But my most important aspect, I'm not too much worried about all those figures. I'll stick to this. And in Northeast India, we have 167 amphibians and 182 reptiles, and this is increasing by leaps and bounces now because we are coming up with lots and lots of new species, and yet we are in a process of describing many. So many of you may not know what amphibians, and I want you to, because I told you I'll give you a very baseline data, not much technical, but many people may have this doubt. Many people confuse between amphibians and reptiles. What are amphibians and what are reptiles? I basically don't like this type of mics. I want those mics which I can travel around. But anyway, I cannot stand in place. Though I work in amphibian reptile, I'm more like having a habit of butterfly. So, if you see amphibians and reptiles, there is a very evolutionary, a very beautiful thing happened. Life started in water and stayed in water till fishes. And after fishes, amphibian went on to the land and they started laying these eggs. Okay, fishes also laid eggs, and the eggs the amphibian laid, the eggs have to be kept moist because they do not have that hard shell. So the eggs have to be kept moist, and that's why amphibian cannot go away from water. They have to be near water, and amphibian itself means biphasic life. Amphibios means two phases of life, one in water and one in land. So their egg has to be laid in water, and the land form would be the adult one. Now, as soon as that happens, they are constrained by the fact that cannot go deep inside the forest or hills because of that problem if there is no water. But reptile, on the other hand, does not have this problem because they are having hard shell, eggs, and they can, all the nutrient and the moistures are inside the egg. So the egg change in the egg, development change the history of the amphibians and reptile and the future groups of animals to come. So please feel free to ask if you have, or you can at least jot down question. I want it to be more interactive. So I'm very happy if you have any in between quick questions. So just to give you, I know these are very boring for people who are not in this field, but my presentation are targeted to the people who are like new in MSc, and they want, maybe they, because there are very handful of researchers working in herpetofauna, amphibians and reptiles. And my goal always is to publicize the topic and get more people involved in the study. So here broadly we have this. How many of you have seen this frog? Raise your hand. How many of you have seen this frog? I see small hands, but I know they can go up little bit more. I see one hand. And what more? Two hands, three hands. Okay, there are more hands. So this is the toad. This is the classical toad, rough skin. There's a total family devoted to that rough skin species. Then after that rough skin species, this is quite human commensal because of it can tolerate wide degree of variation in the environment. So they are more human commensal. Next is there is one more. You can read the details here, but I'm going through the basics of the things. Now the next group of like amphibians or frogs are those which are having not rough skin but moderately rough skin. But the most important thing about these amphibians are they do not jump, they walk. They walk, and this is beautiful to see them walk. So this forms Megophridi. Then Microhylidae is the family where you see there are very small frogs. They are like balloons. They look like balloons, and they are very small frogs. These are Microhylidae. Then Rhacophoridae are the ones which lives on up in the tree. And then finally we have this rennet frog that most of the frogs that we say frogs, not toads. Like generally we say frogs, those are these rennets. Next please. Now this is for students. Oh, sorry, I'm saying next please. I have to do this. Now the most important thing, this frog group are the most problematic group, and those who are working in amphibians and reptiles or frogs, they are having a hard time to decipher how to tell this frog apart, and it will be very useful. So this frog initially was in this group, the last one, rennet, and then finally it was taken out of rennet, and a family was created, Dicroglossidae. Now many times Dicroglossidae people come and ask that how we can identify Dicroglossidae from rennet. I don't know the exact answer, but because it was molecularly done, but there are key pointers. You will, for rennet, you will not get any. All of you listen carefully to the last. Can you hear my voice? Last, can you hear my voice? Sir, tonight there I am. Very good. So, if you see this, the bad. And it will not there. And then, but he's back. The much like this. Now, who can tell me here and no? I can go here, right? Why? You need to know why. You know, it's in the here, bro. Give a try. Give a try. You may be wrong, and that's the beauty. I आता तुम्ही Oh, sorry. So my question is, if the nostrils are here and if the nostrils are here, what difference does it make? कोई भी स्टूडेंट खड़ा हो जाओ यार, बोलो. Tell me. ये they are discussing but still not telling. Come, tell. It may be wrong. You have any idea? Tell. I know it is right. Tell. Huh? Yes, of course, evolution, but there must be some biological implication. Anyway, I think we will get late in time, so we have to hurry up. So, sound is from this part. Not nostril has not much role to play, but biologically, those frogs which live in land, they have this type of nostril because they breathe air. But when you have nostril here, they will come to the surface and the nostril will be at the top. So they will just bring out the nostril and breathe and go back to the air, water. So that is the biological. So if I see nostril here, I can see it's more aquatic group of frog. Okay, these are all the frogs. So I have time constraint. That is the thing. So these are photographs. We can discuss later if anybody is interested. And these are more frogs. These are even more frogs. And these are snakes. Are they snakes? What are they? These are? But why do I discuss fish? I am not discussing fish here. I am discussing amphibians and reptiles. So these are not snakes. Then what it can be? Caecilians, exactly. These are legless amphibians. Amphibians may have legs, may not have legs. Frogs have legs but don't have tail. Caecilians do not have leg at all. Okay? Then I am coming. And that salamander I did not put because yesterday Disempson talked about salamander that we discussed. So reptiles I have already told you about reptiles. Then what are these? Lots of species of turtle. I'll tell you one thing. 21 species of turtle, freshwater turtle and tortoises are found in Northeast India and out of 29 species. So Northeast India is lots of freshwater turtles and tortoises. 21 out of 28 species found in 29 species found in India. So more the number is not only good but also bad because there are a lot species to be lost. So we have to be more careful regarding the turtle. And one turtle which is creating havoc in Northeast India, specifically Assam in Guwahati, is red-eared slider. The pet turtle which is in pet trade introduced, people keep it when they are cute and young. When they become big, they will release in the water and they will compete with our already endangered species of turtles. So we have to stop that. So these are more turtles. This is lizard. And I'll not go in details. Time again is of scarcity. So these are the group of lizard. I'll just tell. Anguidae is a lizard which do not have legs. Then the skinks are the lizard. Do you know skinks? I think you know skinks. Then we have these lizards which is Lacertidae, which is having huge tail. The one of the marker is huge tail. Then Varanidae, which are having Varanus, you know that. Then Agamidae, the calotes and all. And finally the Geckonidae, one of my favorite group, I'll come to that. And then these are other lizards. This is snake, right? No, this is not snake. This is also a lizard. But not having leg does not make it a snake. How snakes and lizards are different, I'll tell you to the next slide. And this here, these are the same species I have taken 10 minutes apart. Calotes Maria joining politics by changing color. So you see, so it was green some time back and it became red suddenly. So I think this is the best candidate. So now one thing I just missed out. You see, in lizards you have ear opening. In snakes you do not have ear opening. Snake cannot hear any airborne sound. These are some of the lizards. This is the beautiful part, the venomous snake pit viper. So I'll not go in details. These are some of the scales of the head of the snake. How does it look? And then this I want to tell, only venomous snake do not only have the fang in the front, they can also have fang at the back. So these are known as back fang snake. This is Rhabdophis hilari, one of the species found in Mizoram as well. So this is the reproductive organ of the snake. We know, we call it hemipenis. These are the thermoreceptor pits. Some of the snakes can see in absolute darkness. Even if you tape their eyes, they can see with the heat sense. They can sense the heat there, and mostly they use their tongue to find prey, because they keep on flickering the tongues, and there is a Jacobson organ at the roof of the tongue. So these are groups of the snake. Typhlopidae are the worm snake, Pythonidae, then Viperidae, Elapidae, and Colubridae. So these are some of the snakes. I have to go fast. And we come to the point, and I am almost done. This is the HT long back when he used to be much thinner. You can recognize it. I have not changed much. Maybe I have lost little bit and HT gained much. So, and the legacy started with HT's lab. And what we did is, we described all those species. So this is one is HT's lab described Tambili. He did not take me as an author, very bad of him. Then this is Ichthyopis benji. Luckily he took me as an author. Trilotreton zynai. He took me as an author. Thank you, HT. And then a scores of 10 species of 110 species of reptile. We described this many species of HT's lab described this many species of Cetrodactylus or geckos. This is the only non-Cetrodactylus. Rest of all are bent-toed geckos, and some more snakes we described together. One or two he did not take, that I have to talk to him why. And then the best part, the last slide I want to tell you, we described so many Cetrodactylus bent-toed gecko from Mizoram. The best part I want to tell you, see biogeography and genetics, how they are linked together. See here, many of you may be knowing this, implication of phylogenetic tree. So how many clades do you see here? This is an outgroup, forget about it. How many clades? You see, broadly I see one, this much, one, this much, and one, that. So I broadly see one clade, two clade, and three clade. Now, there is very interesting biogeographic aspect to this. This is the only species north of Brahmaputra. Rest of all are south of Brahmaputra, and gene also tells the story. Now, in south of Brahmaputra, you get two clades. One is this clade and one is this clade. So all these clades are from Mizoram or high altitude, and these are all from low altitude like Guwahati and other places which have altitude like 300 meters. And these are all like 1000 meters. So also south of Brahmaputra there is two clades which is divided because of low altitude and high altitude. So these are some of the groups of these Cetrodactylus, and they are telling, and I am amazed to know that we have Sri Lankan, fauna in Northeast India. We have genetically we have found three species now, which is the origin is Sri Lanka. And that day I was there in the conference where they are talking about this Ram taking up this, Hanuman taking up this, what is this, sir? Big mountain, Hanuman. Sanjeevani is Bhuti, but that whole mountain, there is a name. Gandhamadan. Gandhamadan Parbat, when they are taking, see sir, it's a mythical story. But they say that when they are taking Gandhamadan Parbat, some of the part, when they are bringing, some of the part of Gandhamadan, it's in Sri Lanka, and some of that fell in Northeast India. And you see how they are correlating with our finding that we are getting Sri Lankan genetic things in Northeast India. We cannot believe, because the species when we sequenced, we thought it will be allied to Southeast Asia. But no, it is fitting with Sri Lanka. So there is magic. So yeah, for our team, the journey has just begun, and we promise to deliver more and more species for this Northeast India. Thank you.
Thank you for the very quick crash course on fauna.
I hope I did not crash.
We have time for one or two very quick questions.
Yes,
Yes.
yes, sir. See, sir, all the this Cetodactylus bent-toed gecko, they are very unique. They are only within. I'll tell you a small thing. Like there was only one species of bent-toed gecko known for all of Northeast India and Myanmar. That was Cetodactylus khasiensis or Khasi Hill bent-toed gecko. Because generalistically everything was put together. Now after dividing, we are getting 38 species out of that one species by because we see now that species, the implication, they are living in small 100 square kilometer, 200 square kilometers patches, not more than them, and they are micro endemic to their habitat. Now if their habitat is destroyed, like in Meghalaya we are trying to work, and in Meghalaya there are limestone quarries where the cement industries are using that limestone. Now in those limestone areas these Cetodactylus are living. Now once that limestone area is being exploited because for cement manufacturing, all those Cetodactylus, before we know, they will vanish because they are living in very small patches.
So, how, when I say that how unique species are that you have discovered in Flora, so does it mean that it is not common anywhere else?
These are new species to science, sir. These are all new species to science. Whatever I have told you, these are all never ever discovered. We are the first team to discover all those 20 or 30 species in this period of 3 years.
So it was never known
No,
in the world.
in the world.
That's good. But then you see, this raises a serious question when you already know the reasons how these species can disappear. So how do you think you'll be able to save them from extinction?
We have no answer to that, sir, because we are giving,
This raises a broad question.
yes, so,
Speak about conserving species.
yes,
How exactly are they going to do it? What are the methods?
see, sir,
Are there unified models or extinction is bound to happen?
I can tell you in details, but as for moment, what I can tell you is, like, take it as an example of bus. Bus driver drives the bus, mechanics take care of the spare parts of the bus, and conductor has his own duty. I am the mechanic of the bus, and I want bus driver to do the rest of the work because I am not expert in that. I am expert in just rectifying the whatever there in the problem in the bus.
So the species that you are telling are unique only to this region. Then there must be efforts made.
The government should make it. Yes, we are too small to make any effort. We can just tell and guide. We can just give them a plan.
This is a platform from where people can recommend this.
So we are recommending, sir, but nothing is happening because we are too much myopic with mammals. We cannot, I say this MMM, mammal myopia, mega mammal myopia. So a place where which is too much obsessed with rhino, nobody, I have been giving chakkar in Babu.
May not be bad to be obsessed with rhino, but the point is when you are only involved in protecting rhino, you should also have.
But nobody cares about us. Our paper do not even fit three or four journal impact. If we get a paper in our 3 to 4 journal impact, we give party because this even new findings. We are the bottom of the strata. Nobody cares about us.
I'm afraid I have to interrupt right now. It's a healthy discussion, and maybe we can have a separate session on conservation, which I'll love to partake. Thank you. Thank you very much.
Thank you, sir. Thank you.
Thank you so much, Dr. Jaditya. In order to show our appreciation, we'd like to present you a certificate of appreciation, and to hand over this certificate, I request Professor Ramesh Pulratna to hand over the certificate.
We now have the second speaker, Dr. Nasimul Hoda, sorry. So, Professor Dr. Hasimul Moda is currently attached with the Department of Chemistry, Jamia Millia Milia Islamia. Wow, that's a mouthful. Alright. So currently his research interest is in uh, organic synthesis and computational biology, dealing with anti-malarial agents, and the first one, organic synthesis, dealing with drug design and computational biology. He has a very long list of publications and a number of research projects that's ongoing, which I'm not going to read out so that he can spend more time enlightening us on his talk. Please, sir.
First of all, good morning, and thank you, Professor Senthil, for having me here. And directly, I'm coming to my talk, and my talk is divided actually in three parts. We do organic synthesis of molecules which are of therapeutic importance. But before doing that, we use computational tools to design those molecules. Later on, after the design and synthesis part, we collaborate with the biologist for the biological screening of our compounds. So some of our compounds are in interesting phase, and we are also looking for the translational output of these molecules. So here, my uh, so here uh, uh, the theme of the work is actually drug design, because we will be utilizing the tools of drug design to design, synthesize, and do biological screening for these compounds who have the potential to disaggregate misfolded proteins. In drug discovery journey, actually drug discovery process itself is a interdisciplinary endeavor which works at the interface of chemistry and biology and utilizes informations or concepts from other field of sciences like computational tools and structural biology, etc., etc., etc. Sorry. Uh, Actually, 2013 Chemistry Nobel Prize was awarded to Martin Karplus and other fellows for the development of multi-scale model to solve complex chemical problems. Actually, this was the founding stone for the utilization of computational tools to solve chemical and biochemical problems. Actually, these tools have applications. These tools have applications in supramolecular chemistry, that is gas post chemistry, as well as in protein folding, and of course in drug design. Actually, we work in protein folding. Actually, my boss used to work in protein folding, and drug design we are working. So their finding is really has helped science to progress in this direction, and that is why it's important to mention their name. So in our work, we will be utilizing, there are several biological computational biology tools to design molecules. Our interest focuses on structure-based drug design. We are just focusing on structure-based drug design to design our molecules. So if one more thing, again, I want to be interactive in the session. If anybody has any problem and some query, something, can interrupt in between and ask the question. So when we talk about the structure-based drug design, where the role of Professor Ashok Verma comes, we know that he is a X-ray crystallographer, an excellent X-ray crystallographer, and his role is important here. So X-ray crystallography is used for getting the structure of the compounds. Then we also depend on cryo-electron microscopy and NMR structures. Mostly we get a static structure from crystallography, and from NMR we get solution structures. And from so these are important. Why structure-based drug design is directly related to a structure? We talk about the guest host chemistry. So just depending on the size of the host heart, or accommodating uh, accommodating room or something, the space available with the host just can occupy. So before that, so what we need to know here is that this is the active site of the protein where the substrate will sit in. So in the first one, it is not having the substrate. In the second one, the substrate is sitting at the active site. So actually, how the substrate binds at the active site of a molecule can be depicted through this structure. This I have borrowed from some others work. Actually, it is of the development of neuraminidase inhibitors. This is the thing, the importance of structure in drug design. So actually, the development of neuraminidase inhibitors. Is an important enzyme, which is an important target enzyme for the development of antiviral molecules, and its substrate is sialic acid. We can see that. Actually, I'm going at the structural level, at the molecular level. I'm sorry for that, but this is my expertise, so I'm dealing with it. I know that in biology, it's difficult for the biologists to, to have understanding. So many of you are good in it, but for many of you, it would be difficult to have molecular level or atomic level understanding. So what actually we are doing here is that you can see that binding pocket information will directly come from the structure of the active site architecture. We have the exact active site architecture with us. Then looking into the binding mode with the substrate of the enzyme, we can look into it, and as some of the speaker was talking about, we need to mimic biosystems. So mimicking is really important. By looking at how nature works, how the enzyme interacts with its substrate, based on these informations. Here you can see that if you have idea about hydrogen bonding, so we can see that the residues which is having guanidine group into it is nicely interacting with the sialic acid through several hydrogen bonds. Here we can, if you want to count, we have 1, 2, 3, 4, 5, 6. 6 strong hydrogen bonds are there between the residues at the active site and the sialic acid, which is substrate for the neuraminidase enzyme. Now, based on these informations, the information was obtained by a X-ray crystallographer. Later on, we can see that a molecule called Relenza was developed. Relenza is actually a success story of computational biologists. We can say that how computational tools and X-ray crystallography tools have been used to bring this molecule into the market as an antiviral molecule. So when you will compare the active site story or binding mode of action of Relenza with that of a sialic acid, you will come to know that they sits well at the active site and they behave almost similarly, and with a few extra interaction between the residue and the substrate. Here you can see that it has 1, 2, 3, 4, 5, 6, 7. 7 hydrogen intermolecular hydrogen bonds are there, and some other favoring factors which counts towards the free energy of complexation. So this has helped to bring this Rilza molecule into the market, which is a favorite antiviral drug. So based on these informations, and what are the factors actually which contributes to the free energy of complexation? So we have just mentioned few. Of course, pi stacking interaction is one of the favorite one. Then hydrogen bonds, then ligand protein flexibility, non-polar contacts, steric clashes. We need to get rid of this steric clashes as much as we can. So now, based on those previous informations, now we will look into the development of our molecules, what we have learned from the history, and how we are going to utilize it to bring our own molecule into the market. And actually, at this point of time, actually it has become a fashion to use bioinformatics tools for the for getting publications and other things. But we want to work into it, and our dream is to bring few molecules into the market for malaria, maybe for neurodegenerative disease, and for tuberculosis. At least we are working in these three areas, and we are sure that one day we will come out with some molecules which would be helpful. Actually, why malaria and tuberculosis? Because this problem is very much our own problem, and we have to solve it. Nobody else will take care of our problem. That is why we have to solve it ourselves. And for that here, actually, because my topic is very much related to the protein disaggregating agents. So in neurodegeneration, as we know that when we are talking about Alzheimer's disease, specifically I am talking about one of the neurodegenerative disease, that is Alzheimer's disease. So here it has a cognitive problem, and with increasing cognitive problem, we know that the memory problem could be enhanced by playing with the neurotransmitters. And acetylcholine is one of the neurotransmitter related to the neurodegenerative disease. So what acetylcholinesterase does with acetylcholine is it hydrolyzes. This is actually cholinergic hypothesis associated with the Alzheimer's disease, where it talks about acetylcholinesterase enzyme hydrolyzes the acetylcholine, which is a neurotransmitter and responsible for the cognitive action of the human system. So what people have done earlier, they have developed acetylcholinesterase inhibitor so that the acetylcholine level is maintained in the brain, and with a greater level of acetylcholine, the cognitive problem could be taken care of at least for some time. It is not a permanent solution, but it doesn't have disease modifying capability, but certainly it helps in improving the cognitive behavior of a person. So this is one thing. So we have to focus on an enzyme which is called acetylcholinesterase, which we have focused on, and it is really an interesting story, and how we can use these informations, the stepwise approach, to do our basic science, and which can be later on transformed into translational work. And the other approach is, as we know that this Alzheimer's disease is also associated with the extracellular deposition of amyloid plaques in the brain. Actually, I have not gone into details of all those things. Just I am focusing on few of our research work. And this is amyloid hypothesis. As earlier I told, this Alzheimer's disease or most of the neuro disease have multifactorial approach. We need multifactorial approach to take care of it. And in our actually amyloid hypothesis. Actually, we have amyloid beta 1 to 42. Amyloid beta is there 42, 43, and this aggregates and which results into the formation of the extracellular plaques in the brain. So we have to get rid of those extracellular plaques, as well as at the same time we need some approach which can also inhibit the enzyme acetylcholinesterase. So here we have used two-pronged approach to solve this problem, and we are trying it just we are proposing, and we have some success into it. So this is actually when actually I'm not I have some maybe this. So actually this is the active site of acetylcholinesterase enzyme. You can see that it is a narrow Golgi. Narrow Golgi is there. It is actually 20 angstrom Golgi from the opening of the enzyme phase to the end of and the catalytic site is sitting at the bottom of the Golgi, and it has one more active site present at the opening of the Golgi. And when you will look at it, it is a serine hydrolase class of enzyme, where serine 200 is there, histidine 440, and tryptophan 84 is there at the active site. These three amino acids are really involved in the hydrolysis of the acetylcholine at the bottom. But it is sitting at the end of the molecule, and the acetylcholine molecule has to come all the way through the opening at the bottom of the enzyme. During the process, what happens is, if the next stage, this is actually obtained from the crystal structure. It has been shown that tryptophan, which is present at the 279 tryptophan, which is present at the opening of the Golgi, acts as an anchor and as a director. What it does is, it invites the acetylcholine towards itself, and then it directs the acetylcholine towards the active site or the catalytic site of the enzyme. In this process, when you will look at this, actually tryptophan is having an indole ring. Indole ring, flat, bicyclic, heterocyclic ring. Flat ring, which is aromatic. Aromatic rings, what are the properties of the aromatic rings? Aromatic ring has delocalized pi electron cloud associated with it. Because of that delocalized pi electron cloud, it interacts with cations, as well as we have learned from the nucleic acids, the flat heterocyclic rings, they are very much capable of stacking with the other flat rings. So these informations from the structural organic chemistry could be utilized here to design new molecules. Yes, as we look at the thioacetylcholine, actually this is the thioacetylcholine which is interacting with the tryptophan 279. We can see that they are interacting because the thioacetylcholine or acetylcholine is a cationic species, and here they are interacting with each other through pi cation interaction. And later on, this is our work, and later on, a molecule called donepezil. Donepezil is considered as a dual inhibitor of the enzyme acetylcholinesterase. What it does is, this is the actually not able to use it. So this is actually the donepezil molecule, which is binding at the active site, and it is simultaneously interacting with the catalytic anionic site and peripheral anionic site. It is interacting with both the sites simultaneously, and designing a molecule which can interact with both the sites is challenging. Why it is challenging? Because they are at two ends, and a very narrow Golgi, aromatic Golgi, separates. To overcome this, actually we have tried. This is actually donepezil is the FDA approved marketed drug to treat Alzheimer's disease. So this is the crystal structure, and based on these informations, we have designed our molecule. When you will look at this crystal structure, you will come to know that the tryptophan 284 is having pi stacking interaction with one of the aromatic ring of donepezil. Simultaneously, the other tryptophan 279, which is also interacting with the bicyclic aromatic system at the bottom of the Golgi. So these informations and the exact measurement between these two sites, which is around 20 angstrom. So based on these informations, we designed our own molecule, and actually this work was done by Dr. Jitender, and right now he is assistant professor. So he did all the synthetic work, and together we did the actually biological and for computational biology tools, we also use Professor Jairam's lab at IIT Delhi. So we did this, and when you look at actually that one is the docking study followed by molecular dynamic simulations. So when you will look at that structure, you will come to know that the way donepezil is sitting at the active site, similarly our molecule is also sitting at the active site. Previously we designed it and then we synthesized it in our lab. So when we are doing some bioinformatics work, this is a message for our young researchers who want to have their career in research and drug discovery, or one who is doing bioinformatics work. Please integrate your bioinformatics finding with experiments. Please integrate it. Otherwise, it is of no use. So, first of all, when you are designing a molecule, you must have idea about the synthesizability of the molecule, whether we can synthesize those molecules in lab or not. This is really important. So what we do is actually we only design those molecules which we can synthesize in our lab, and at least our lab is well equipped in such a fashion that we can synthesize any synthesizable molecule on. If somebody else could synthesize anywhere in India or somewhere, we can also synthesize those molecules in our lab. So, and we do not focus on methodology development. We focus on total synthesis. Okay, you give the target, you give the molecule, we will synthesize. But it should have synthetic potential. It must be synthesizable. If it is not synthesizable, we cannot. So before going into the bioinformatics work, you must have idea which molecules are synthesizable, which molecules are not. And although we did this work in 2018, but I am still proud of this work because it has paved the way for better understanding and the development of certain exciting molecules. This is second work based on actually Dr. Mudassir and Dr. Ehtesham. Dr. Mudassir is presently is in USA, NIH, National Institute of Health, and Dr. Ehtesham right now he is in CDRI. He is senior research associate at CDRI. Right. So same work, and you could look at the structure of the molecule, which we actually it is a bicyclic system, triazolopyrimidine kind of molecule, which is connected with the triazine, and the molecule is linked through piperazine moiety. Pardon?
You have 2 more minutes, please.
Just 2 minutes. Okay, fine. So, and later on, actually, now we are reaching to our beta amyloid disaggregating capability. So, first of all, our aim was a molecule should have two properties. The first property was associated with inhibiting the enzyme acetylcholinesterase, and the second one is to disaggregate the misfolded aggregated proteins. So in that, we did this TEM experiment is shown here, and our molecules are better beta amyloid disaggregator than the well-known disaggregator, which is curcumin. With respect, the first that one is with curcumin, and these studies are with our molecules, and these studies are superior to that of the curcumin. And later on, actually, this is another structure here. Here, actually, this design. So we designed the hypothesis. We proposed a design hypothesis. How we have designed a molecule which is capable of disaggregating the aggregated proteins. So for that, actually, the role of a proline is well known in literature, which suggests that uh, it is a beta sheet disaggregator. And what we did, we incorporated this proline, then piperazine, which is having a puckered structure, and then planar systems. Planar systems works as a sharp knife, and this puckered system of proline, which is also having non-planar part, and the piperazine thing. Actually, they help to disaggregate things. First, it embeds into the sheet, and then it disaggregates it. And for that, something else needs to be modified for the better hypothesis where we are working upon. So these are few results. Again, we are also working on some fluorescent probes, but because of paucity of time, I am not doing it. And this is Shoaib Manzoor, another student of mine, which is right now he is in University of Bath and planning to, again, he is also planning to visit an IH lab at there. But just I'm wrapping up, and at the so this is another example, and this is recent work. Recently we have published, and a very good work. And now this Kavita, Hari Madhav. So these things I am not going to mention. Yeah. So actually we have utilized the focus on few things. What I had seen in Western world, if they don't focus on many things simultaneously, they focus on very few things at one time. Later on, they integrate them together. So do like that. So these are the summary and takeaway message. And most importantly, I would like to acknowledge our collaborators, because you know that if we don't do biological screening, and we are always looking into some collaborative venture where we can collaborate with biologists of our interest. So I am with Professor Mizoram. He is like a father figure or mentor for me, and Professor Dr. Pawan Mohatra from ICTEB. He is actually a collaborator for doing anti-malarial activity, and Shobik Bhattacharya from JNU, Amir Nazir, who did from CBI, he did some C. elegans model work we did with him, and then Sharad Gupta from IIT Gandhinagar, again alpha synuclein activity, and Philip Rosenthal from University of California for anti-malarial activity. And funding agencies, ICMR, Spark I am having, and of course DVT with Professor Ashok Verma and Senthil.
All right. Thank you, Professor Hoda.
And this is my great.
Let's give him a big hand. I'm afraid we have no time for any questions, but for those of you who are interested to ask him and engage him, I'm sure he'll be available at other times to discuss. Sorry, no question, please. You want a question? Okay. Sorry, there's a request for one question.
Good introduction to here, how to start things on drug designing. You see that steel orient structure, you show the active site as a gorge, right? And the chemicals that are already used or the ones that you have synthesized, does it interact with any other site when the circuit before it enters into the active site? That conversion will go because we have not yet hypothesized the molecule. Right? We will touch first time before. And we are looking for collaboration. Hopefully, if Professor Ashok Verma will be kind enough and very great with it. But we are, right now, we have shifted from this level. Actually, why I'm talking about this story is it tells us about the way our research work can proceed. Yes. Or use a drug design tools to design more and go up the end of. Actually, finally, is there a relationship? Is there a relationship between acetate choline pathways that you talked about and the amyloid? Are those somehow connected or they work in different substance? There is another ionic site that is hypofactive 79. It interacts, actually, it acts as a basic. So it has this capable of attracting amyloid beta toxicity. Okay. So it has. So it is a method where amyloids are a mixture of various proteins. Amyloids are a mixture of various proteins. So another issue, amyloids are mixture of various proteins of actually it is there is an amyloid precursor protein which is cleaved and varies by several proteins. And after that, it is the byproduct of AFI. It is the byproduct of AFI. So absolutely sure, yes, we are absolutely sure that it comes from the amyloid beta aggregates. Yeah, it's a good. Maybe 42 among the 6 are there, 43, 44, maybe good. Oh, thank you, sorry. And there is one thing related to the multiple active site. No. It is a flexible protein. Research acetylcholine is a flexible protein and may bind at others. Live-river workflow, as suggested us. It is binding at more than one. More, yes. Thank you.
Thank you so much, Dr. Nasimul Hoda. I now request Dr. Esther to present Dr. Nasimul with a certificate of appreciation. Once again, I would like to thank both our speakers this morning, Dr. Jayaditya and Dr. Nasimul. Also, I would like to thank our session chair and co-chair, Professor Ramesh and Dr. Esther, for guiding us through this session. As Indians, we sure love our chai. So, ladies and gentlemen, may we spare 15 minutes short tea break, after which we will have our next plenary session. Meet you promptly after 15 minutes.